Okay, and as we continue to wait, I just want to remind everybody that we have our monthly journal club today from 4 to 5. And we're in person, the third floor Semel conference room. And we're also over Zoom. So if you want to attend our monthly journal club and you have not gotten a notification, you can email me. You can't, chat is disabled, so you can't do that, but you can email me or email cannabis@mednet.ucla.edu and we can send you that information. Just as a reminder, we have these monthly journal clubs. They're geared towards really all types of cannabis science. You can bring in a journal club, a journal article of interest that you want to discuss. You can bring in new data. If you're giving a talk and you want to practice, you can do that as well. We just like to plan about a month or a month and a half ahead of time. So just let us know. It's really geared towards trainees and junior faculty, but everybody is welcome to come. And we're hoping that this will help to kind of continue to foster a multidisciplinary dialogue across our groups, across our departments and divisions. So we're really excited. Connor Murray is really taking a lead in a postdoctoral fellow in our lab has really taken a lead in organizing it. We've already had a couple of sessions, so it's been really exciting. Other announcements, CPDD is happening tomorrow. Let us know if you're there in Denver, starting on Saturday to next week, and then the International Cannabinoid Research Society meetings happening the week after in Toronto. So please let us know if you'll be attending. We look forward to seeing everybody. And on that note, I am so happy to welcome Dr. Jodi Gilman, who's an associate professor in psychiatry at Harvard Medical School and the director of neuroscience at the Massachusetts General Hospital Center for Addiction Medicine. She received her Ph.D. from Brown, where she participated in the graduate partnership program with the National Institute on Alcohol Abuse and Alcoholism. Her research uses multimodal imaging, behavioral, and cognitive testing to understand the biological, psychological, and clinical aspects of substance use disorders. And she's specifically interested in studies that look at the effects of cannabis use on substance use and health outcomes. And really, that's what she's here to talk to us about. And her overarching goal of her research program is to conduct research that can inform decision-making among the public, patients, and clinicians regarding evidence-based decisions to use or not use cannabis for medical or non-medical purposes. She's authored a series of scientific publications on the acute and long-term effects of cannabis use on the structure and function of the brain, specifically in young adults. And she's also co-authored book chapters on the connection between cannabis use and mental health outcomes. She's all over the media. You know, if you're in your car on a Sunday and you hear, this big science clips on NPR, frequently you'll hear about Jodi's work. She's been on the BBC and Reuters. So it's really wonderful to have Jodi here. And personally, on a personal note, Jodi and I started working together this past year with a couple of other people here at UCLA, including Marjan Javenbach and Lillian Gelberg, and it's been a real pleasure to work with Jodi. So it's wonderful to have her here. Just a reminder, when you have questions, please put it in the Q&A. We've already received some of the questions, so we'll be able to ask those at the end. And welcome, Dr. Gilman. You're here to talk about recreational medical cannabis, potential implications on cognition and clinical outcomes. Thank you.
Thank you so much for that nice introduction. I'm so happy that you invited me to talk to your group today, which just sounds like such a great group. And I echo everything back to you, Ziva, how much of a pleasure it's been to work with you this past year on some grant submissions and things. So thank you for inviting me. And as mentioned, the title of my talk is Recreational and Medical Cannabis, Potential Implications on Cognition and Clinical Outcomes. And sadly, no one is paying me to say any of this. So this is all, just my scientific work. So I'd like to talk to you about a few studies that we've done in the past few years regarding cannabis, starting with some background about cannabis in the brain, which I'll keep very brief because this group is probably among the most knowledgeable in the world about cannabis in the brain. And I'll talk to you about some studies that we've run on cannabis and cognition in adolescents and young adults. A study that was just published actually a few months ago in JAMA Network Open, which was an RCT, a randomized clinical trial of medical marijuana cardholders. And then I'd like to talk to you about some of my neuroscience work addressing cannabis and driving, as well as some future directions. That's a lot in 40 minutes. I may get through it all, I may not, that's okay. But I figure I could try to share as much as I can. Okay, so I never know how to describe cannabis anymore. I'm just going to keep this slide up. Cannabis is the most commonly quote unquote illicit drug in the US. It is still federally illicit. It is now legal for medical purposes in all but 8 states. So we've really changed the policy landscape dramatically in the past decade. And its point is it's widely used. Over 100 million Americans have tried cannabis, and 2.5 million people used it for the first time in 2020. So cannabis use is just growing dramatically in the United States. This is a map of the distribution of THC binding sites in the brain. And as you can see, THC binding sites are really ubiquitous throughout the brain, which is why when somebody uses cannabis, it affects a variety of domains. There are THC binding sites in the basal ganglia, which is involved in motor control and planning, in the amygdala, which is responsible for anxiety, emotion, fear, the neocortex, which is where a lot of our cognitive functioning lies, hippocampus, and cerebellum, so memory, motor. So really, and anybody who has used cannabis would know this, cannabis affects all these different aspects of the brain. The other thing that I need to say before I talk about, before I give any cannabis talk, is that cannabis has changed dramatically in the past decade or so. So cannabis in the 1960s to the 2000s was basically leaf-based cannabis, THC concentrations between 1% and 3%. You'd get a little high. And quite frankly, when people voted for legalization of cannabis, this is the cannabis that a lot of people had in their mind. But what's going on today is quite different. So cannabis is not just a plant anymore. These derivatives of cannabis can contain up to 98% THC. We have waxes and oils and shatters and butters and all these different products, and new forms of THC delivery, like this little jewel pod where you can put THC oil. And this really changes the landscape of cannabis use, especially among young people. We do a lot of school-based studies, and what we're told is that kids can be high all day long in school and nobody would know. There's no smoke, there's no odor. They can put this USBs, they can put it under their sleeve and just use it all day long. And these are quite potent products. That's not even to say get into the edibles. So now there's a thriving edible market, particularly in states like mine and California, my state of Massachusetts, where this is all legal. You can find this anywhere. It's easy to get and it's easy to hide. So if you can imagine, you take these things out of the box and you put them in your lunchbox, and nobody would ever know that you're using cannabis during the day. Now, I just want to make this point. Although the title of my talk is Recreational and Medical Cannabis Use, I would like to argue that this is really a spectrum. So you can imagine, people using cannabis for recreational purposes. They like getting high with their friends. People using for medical purposes, they want their pain to feel better. They don't want to get high. But you really have a lot of people sort of in the middle saying things like, it helps me relax, or it takes the edge off my pain. It's still there, but I can get a little stoned and it helps. So I have stopped really designating between medical and recreational use because I really think there's a ton of overlap in the population. And now I just call it cannabis use. And I'll get into that a little bit more later. So the first study I'd like to show you is a study on the effects of cannabis and cognition in adolescents. So this has been known for a very long time that adolescence is a critical time in brain development. And there's developmental changes that are ongoing into the third decade of life in connectivity and chemistry and morphology. And if you look at this very classic figure of brain development, and this is gray matter, you can see that the brain develops from back to front, and the prefrontal cortex is sort of the last region to quote unquote fully develop. And what we think is happening with cannabis is that this ongoing brain development may magnify cannabis effects. And this has been shown in animal studies where you can administer THC to an adolescent rat versus an adult rat, and the adolescent rat would show more worsened cognition. So this is a study that's somewhat old now, but I think it makes a really important point. We brought people in who are cannabis users or healthy controls who weren't cannabis users, and we gave them a memory test. And this is a really simple word learning task where we read them a list of 20 words, and then they have to read them back to us. And then we read them the list of 20 words again, they read them back to us again, and each time they get better and better. And then there's a delay period, and at the end of the delay, they have to once again read us the words. So what has been well known in the literature is that cannabis use is associated with poor memory. And when we think of memory, we think of recall. So we think about the data here, where you say, I give you a list of words, and 20 minutes later, you have to recall them. But what we found in this study, which is, I think, a really important finding, is that the cannabis users showed weaknesses in initial encoding or learning. So they remembered everything, and their rate of learning was almost identical to the non-users. And sorry, I didn't explain this very well. This is the group who started using cannabis early, before the age of 16. This is a group who started using cannabis after the age of 16, and these are healthy controls. And what we see is that these early onset cannabis users had trouble learning in trial 1. So they just weren't learning the material in the first place. It wasn't that they were forgetting it later. There were no differences in percent retention, but the early users of cannabis used fewer overall learning strategies. So this is something that has now been replicated a number of times, and it has implications for adolescents using cannabis, I think. The other thing that we found is that this was even worse in kids with psychiatric diagnoses. So in this study, we brought in a group of adolescents who were healthy controls, cannabis users, adolescents who had major depressive disorder, and then the comorbidity, people with major depressive disorder who also use cannabis. And this is the total words remembered in each trial. And as you can see, there's this dose dependency where the people who used cannabis and had depression showed worse outcomes than any of the others. So it was additive. And when you look at the short and long delay, and that's when you ask them, 20 minutes later or an hour later to repeat the list of words, then you really see this difference. So we think that not only does cannabis, interfere with the learning process, but this is even worse among people with psychiatric comorbidities. So the really critical question that I'm asked a lot is, are cognitive deficits reversible? So let's say you have a kid, they're smoking a lot of pot, maybe they're not doing as well in school, but what happens when they stop? Do they go back to normal? So we did this clinical trial of abstinence in adolescents, where we used something called contingency management to incentivize cannabis users to stop using cannabis for a specified period of time. Contingency management is just a fancy way of saying we pay them to stop using, and they get paid more and more and more the longer they stop using. So it becomes, they have greater incentive to maintain their abstinence over time because the payments keep escalating. And what we did was we measured changes in cognition with abstinence. And this is important because it was a randomized trial. So other studies have just looked at people when they stopped using and measured cognition. But here we randomized people to either stop using or continue use. Or I should say, don't change their behavior. We didn't tell anyone go use cannabis. So this was a study of high school and college students. They all used cannabis weekly or more, ages 16 to 25. They were not seeking treatment, so they were all very happy with their cannabis use. And we randomized them at the end of baseline visit. And the randomization was either to the abstinence group, where they were asked to stop using cannabis for 1 month, and they provided urine samples to verify this, or they were assigned to this monitoring group where we said, don't change your behavior on account of this study, just do what you're going to do. And our outcome measures were cognitive. We measured cognition with something called the CanTab neuropsych battery. And the two domains that we were interested in were attention and memory. And we had a few tasks of each, and we did a PCA so we could get a good measure of each domain. And let me show you the results. So first of all, kids will stop using cannabis if you pay them enough. This was great. We didn't know if this would be true or not, but these are carboxy THC levels in urine, and this is baseline and throughout the trial. And this is the group that was assigned to abstinence, and this is the continued use group, a lot more variation, of course. So most of them, more than 90%, were able to stop using for 4 weeks, which was cool. So what happened to their cognition? So first, attention. We saw nothing with attention. So attention got better every week. This was a very traditional practice effect. They were given the same task week after week, and both the abstinence group and the monitoring control did the same in attention. But with memory, we saw something strikingly different. So the abstinent group, so they were all using at baseline. In 1 week of abstinence, they improved their scores, and they basically stayed there at that level for the remainder of the trial. Whereas the monitoring group didn't benefit from practice at all. So their scores were pretty flat. And as you can see, the differences between these groups were highly significant in this trial. So what could we say from this trial? So memory improved among the adolescents who abstained from cannabis, but not among those who continued use. And we didn't see this with attention. This finding is consistent with other studies showing that neurocognitive dysfunction persists after several days of abstinence, particularly with memory. And this appeared to happen within 1 week of continuous cannabis abstinence. So that was actually pretty promising from sort of a public health perspective. And the study provides evidence that adolescents and young adults might experience improvements in their ability to learn new information when they stop using cannabis, which may seem obvious to a lot of us in this field. But, when I talk to, parents and teachers and kids, and a lot of them don't think cannabis is affecting their ability to learn, but this trial shows that it is. Okay, so based on this cannabis work, this was all in adolescence, and I began to become more interested in sort of medical cannabis use. So we can say, this might be true in kids using cannabis for fun, but what about cannabis for medical symptoms? Are these people different in some way? And that brings me back to one of the first slides of the talk, which indicates that they're probably not all that different. So I don't need to tell this group, this background information, but just so we're all on the same page, medical cannabis products from dispensaries are not FDA approved, and FDA approval is an extremely important part of our medical process in ensuring that medications are effective and safe and properly labeled. And the FDA can't evaluate medical cannabis because it's a plant. It's not a standardized formulation, and medical cannabis is different everywhere, depending on how it's bred, the conditions that it's grown, and a million other factors. So you can't be confident that what you're getting is the same from batch to batch, from state to state, dispensary to dispensary, and so forth. And commercial cannabis just hasn't been tested to medicinal standards. It may very well have therapeutic properties that deserves to be studied. And I know that Dr. Cooper and a lot of us are studying this. But the truth is that for commercial cannabis, double-blind placebo-controlled trials just don't exist right now. And when there have been trials for specific cannabinoids, a lot of them have been small and somewhat unsuccessful. So not to start out with a bummer, but the point is that medical cannabis is not often treated like a medicine in our society. So this is something that came into my email box a few weeks ago. It was a Groupon for a medical marijuana certificate. No appointment needed, quick and easy, get certified online. That is not how we treat medicines. If you can imagine, we'll do a thought experiment that this came through for Prozac. Get your antidepressant online, no appointment needed, quick and easy. We'd be up in arms because it's not safe. So I think it's important to sort of realize that while we can talk about cannabis as a medicine and we can test it for specific medical conditions, it's really outside of traditional, practices of medicine, especially when you're sending these Groupons to people. So how do we test efficacy of dispensary cannabis as a medicine, given all of these limitations? So some trials have used standardized formulations like dronabinol, which is synthetic THC, or Epidiolex, which is a FDA-approved CBD medication, with known doses of regulated THC and CBD, as with all traditional trials. So you can do that. But the criticism of these trials, and I know because I've done them, is this is not what people are using in the real world. People aren't using dronabinol in the real world. So saying dronabinol works or doesn't work isn't relevant to this conversation, is the argument. And a traditional RCT would inform us about the biology, but it would not test the system we have in place for the regulation and distribution of medical cannabis. However, most pragmatic trials, so you can say, well, you can just follow people who are using medical cannabis, and some people, including some in this group, have done that, and that's really cool. But there's limitations to that too, in that those trials generally don't have a control group, which makes results hard to interpret. As we know from antidepressant trials, usually just being in a trial makes people feel better. Coming in and talking about their symptoms and focusing on their symptoms can be associated with some improvement. So we designed sort of this naturalistic, pragmatic RCT, and we randomized people to either get a medical marijuana card or be in a waitlist control group. So in this trial, and this was my first R1, and I didn't know if it would get killed at study section, but they actually really liked it. We were looking for people age 18 to 65 who are seeking a medical marijuana card for pain, insomnia, depression, or anxiety. The reason I picked those four is those are the four most commonly used indications, and they're extremely comorbid. We excluded, and this is really critical, we excluded for daily cannabis use or cannabis use disorder at screening or baseline, and for major medical illnesses like cancer or psychosis and major SUD. In keeping with the pragmatic nature of the trial, participants who were assigned to the medical marijuana card group were responsible for arranging for and paying the cost of obtaining the cards and buying the cannabis products. We didn't give out cards, we didn't give out products. So we were really emulating what would happen in the real world when a patient decided to get a medical marijuana card. And I should mention, this was before there was recreational cannabis in Massachusetts. That happened in the middle of this trial, really screwed it up, but that was beyond our control. And so particularly in the beginning of this trial, everyone who was using cannabis needed to get a state issued card. It didn't really screw it up. It was just made recruitment a little harder. So this is sort of the flow diagram. So we had a screening visit, and then people were randomized to an active study group or a waitlist control group. The active study group was allowed to get their card right away. The waitlist group agreed to wait 3 months, or 12 weeks rather, I should say, to get their card. And then they were assessed at the same time points as the active study group. And then there was a 6 and a 12-month follow-up visit after the end of the RCT. We wanted to look at 6 and 12-month outcomes, but we didn't think people would wait 6 or 12 months to get a card if they really wanted one. So we had to sort of balance those priorities. They did a brain imaging assessment at baseline and 12 months. And the outcomes were changes in cannabis use disorder and changes in their symptoms of pain, anxiety, depression, and insomnia. And the first 3 months of the trial, so in the randomized period, we did daily diaries of cannabis use. So every day they would log in and say how much cannabis they used, and they would log their symptoms every day, their symptoms of mood, pain, anxiety, and sleep quality every single day. And the reason for that is because we know there's a lot of variation in these measures. So instead of just assessing them 4 times, we wanted to look at daily variations. Okay, so if anyone wants to identify a problem with this trial, an obvious one is we don't know what these people are taking, right? I just gave you this whole spiel about we don't know what's in cannabis and it varies. So instead of testing products, which is, there's a lot of regulatory hurdles, we tested urine. So we took urine samples, we sent them off to the University of Colorado, and we were able to do an assay for 11 different cannabinoids that are in urine. So we could see what were these people taking. Okay, so just to review the outcomes, one of the outcomes was cannabis use itself. This was such a new field at the time. We just wanted to see what sort of patterns emerged when people started using medical cannabis. Cannabis use disorder, of course, symptoms of scores of pain, insomnia, depression, and anxiety. We had cognitive outcomes, and then we had our MRI and fMRI measures of reward, inhibitory control, and memory. This was our study flow diagram. So we randomized 2-to-1 to card versus waiting list. And the reason for that is a lot of people randomized to getting the card didn't end up getting the card. They changed their mind. It was too expensive. It was too hard to find a doctor. I don't know why, because it seems like they're everywhere. A lot of them just changed their mind. A lot of them just decided they didn't want to. So in order to have roughly equal groups, we ended up doing a 2-to-1 randomization. Okay. So this was not a very diverse sample, unfortunately, as far as race or ethnicity. These were people mostly in their late 30s, early 40s, majority female, so about 65% female, and majority were white. So over 80% of them were white, and they were highly educated. Most of them had some college or a 4-year college or even graduate school. So this was a white, highly educated female group. And that's a limitation of this study that I just need to put out there, but we really tried. We really tried to recruit a diverse sample, and it wasn't easy. The primary, about half of them used cannabis more than weekly when they started. So remember, daily cannabis use and CUD was exclusionary, but we couldn't get totally cannabis naive people because people who want to get a card are generally people who do have some experience using it. And their primary concerns were pain, about 30%, insomnia about 20%, anxiety or depression around 44%. Okay, so cannabis use patterns. So number one, the randomization worked. So again, we weren't telling people what to do. We were just saying you get a card and you don't. So even the waitlist people, we didn't say don't use cannabis. We say, are you willing to wait to get a cannabis card? And what we saw is that the people in the waitlist group generally use less than once a week to less than twice a month throughout the trial. So they were very light users. And the people who were assigned to the card pretty quickly started using around 3 to 4 days a week. Some of them used daily. So the randomization worked quite nicely. So this was our most striking result. So for cannabis use disorder, and remember, this was exclusionary at baseline. The medical cannabis group had 3 times the odds of a CUD diagnosis in this trial as the waitlist group. And what you can see from this graph is this is almost entirely driven by the depression and anxiety group. So remember, we talked about comorbidity earlier. Basically, what this indicates is that people who have co-occurring mental health disorders tended to have more problems with their cannabis use. So this, the people in the pain and the insomnia group didn't really show a whole lot of CUD, but the people in the depression and anxiety group did. The most common CUD symptoms were tolerance and continued use despite problems with use. So some of them will say, I don't love the way it makes me feel, or, I have a headache the next day, but they continue to use. So let's talk about the effect on symptoms. So we saw worsening of CUD, which I showed you in the previous slide. We showed improvement, significant improvement in insomnia. So people were able to sleep better. And we saw absolutely no difference between waitlist control and medical cannabis group in pain, pain, depression, or anxiety. And depression and anxiety in particular is important because these are the people who were developing problems. So not only were they developing problematic use, but it wasn't really helping their symptoms in this group. And pain I was surprised about, but that was totally flat. We found really no improvement in pain between the 2 groups. Okay, so remember we had these daily diaries. So these daily diaries, let me take a minute to explain this slide. So this was sleep quality, Z-scored. And on the left, we have non-use days, and on the right, we have use days. And they rated their sleep quality. So all participants showed an improvement in sleep between non-use days and use days. Even the mood group, the anxiety and depression group, showed a slight bump in their sleep quality, although their sleep quality wasn't so bad. And then you can see the insomnia group These were non-use days and these were use days. So they had a bump in their sleep quality, although they still were reporting lower than average sleep quality because they still had insomnia. And then pain, we didn't see a difference. So let's compare that to, and this is just showing that days since baseline in the medical marijuana card group, sleep improved, whereas waitlist group, it didn't. And this is an all participant, and this is by presenting problem. And you can see that the sleep group is really driving this improvement. So I believe this outcome. I believe that people were getting better sleep. And I also believe that for mood and pain, nothing really was going on. So you can compare that to their pain rating in each group. So this is pain rating in the whole sample, and this is pain rating in the mood group, the insomnia group, and the pain group. Really not much going on here. And then these were depression scores in everybody. Now the mood group, there was I guess, slight improvement, but these are highly overlapping bars. This was not significant. And again, sleep and pain group, we didn't see a difference. Cognition, no differences in cognition. So the waitlist group versus the cannabis card group didn't show any cognitive differences. So this could be also a difference between adolescent users and older users. As I mentioned earlier, a lot of the cognitive effects are seen in younger people. So that's good news for people who are interested in using cannabis medically, that we don't really see cognitive effects. We also didn't see brain changes. So this is an NBAC task, which is a memory task. This is two conditions. One is a 2-back where you have to use working memory, and a 0-back, which is just an attention task. And the expected parts of the brain are activated, and we see no differences between baseline and 1 year. So that's some more good news. And some more good news is that adverse events were generally mild. We didn't have any adverse events related to psychotic symptoms, mania, hypomania, worse cognition, suicidal ideation. We had reports of transient AEs, nausea, dizziness, and paranoia, but nothing that I would consider super serious. So that was good. Now, remember I told you we had this outcome at 6 and 12 months. So the interesting thing here is that by the 12-month time point, use had really converged. So this is the 3-month RCT, and you can see the groups were quite different. And then after 3 months, we just did an observational cohort study. We just followed them for a year. Medical cannabis group use went down a little, waitlist control group use went up a little, and they ended up converging. And then at the end, we looked at CUD, and about 11.7% of all participants and 17.1% of daily users developed CUD in the course of this trial. That is not all that different than quote unquote recreational use. So when we talk about, are people using medically different in some way? I'd like to argue, no, it's the same drug. Motivation for use, whether medical or to quote unquote get high, doesn't seem to be protective in CUD. Okay, so what are risk factors for CUD? So what we did in this paper is we looked at patterns of use. And here we just looked at 3 to 12 months because we thought that, the 0 to 3 month, they were adjusting, they were experimenting, they were trying to get, their use, find their pattern. And we looked at different patterns of use, doing a trajectory analysis. And we found that people were generally in 5 patterns: a low, stable, or no use, medium use, high, stable use, de-escalating. So they started using more and ended up using less, or escalating. They started using a little, ended up using more. And we found that the people who were at risk for CUD were this escalating use group, more so than any of the other groups. Almost 30% of them ended up developing CUD. So for clinicians in the group, if you have patients using cannabis for medical symptoms, escalating use is something that we think you should pay attention to. Okay, so why wasn't cannabis effective? There's a few possibilities. Maybe cannabis simply isn't effective for pain or mood symptoms. And a lot of RCTs haven't been significant for these. So, maybe that's just the way it is. Or maybe participants weren't receiving the right products. Maybe there was lack of regulation, there was mislabeling, they weren't using what they should have been using. Or maybe participants weren't receiving proper guidance on what to buy. A lot of participants asked me, what should I get? I don't know. So they would ask somebody working at a dispensary, and they would make all kinds of recommendations, but these people have no medical training. So there's just not good evidence on what products or doses work for specific medical symptoms. So we addressed two of these. So first, we looked at the metabolites in urine. So at each visit, the participants provided a urine sample. They reported their recency of use, whether their primary products were THC dominant, CBD dominant, or equal. They've all used for the urine analysis that we did within the prior 48 hours and at least 3 to 4 times a week. And we tested for 17 metabolites. So we found that, buyer beware, we found that a lot of people, their urine didn't match their self-report. So among people who reported CBD dominant products, there was no detectable CBD in 30% of samples. THC was detected in almost 80% of samples from participants reporting use of CBD dominant products, which is okay because CBD dominant could mean there's THC in it, but it was just surprising that more of these samples had THC than CBD. And among people reporting THC dominant or equal CBD THC products, no THC was present in 11% or 35% of these samples respectively. Now, vaping was the worst culprit of all. Vaping was the most common method of administration, but 20% of samples contained no measurable cannabinoid whatsoever. So what we did was reanalyze the entire study only with the people with positive urine screens and only in the weightless control people without positive urine screens, so we could have more pure groups, and the results didn't change a bit, like nothing. So we have that in the supplemental section of the paper, and the estimates are almost identical. Poor regulation is a problem, and I just want to read this to you because this is, pretty scary. I was taking what I thought was CBD oil, and apparently it wasn't what I thought it was. I started feeling the effects when I was driving, which was really scary. I got home as quickly as I could. I felt so high, I didn't know where I was and couldn't focus. And the only way I got home was the noises from GPS. I was paranoid that I might have hit someone or something, but I checked my car and there was no damage. So this was a woman who didn't have a lot of experience with cannabis and thought she was taking CBD oil, and clearly she wasn't. So people think of cannabis as something, harmless. And I guess this trial shows it's not worsening anything. Generally pain, mood, anxiety isn't getting worse. It's just not getting better. Although we do show the development of CUD, but generally it was mild CUD. But stories like this, indicate to me that this is not, there is risk to this. This is a psychoactive substance. It should be taken seriously. Okay. Were patients just getting poor information? So this is a density plot, and we surveyed people about how helpful they found the doctor who gave them the cannabis card or the dispensary. And people generally found the dispensary more helpful than the doctor, but not terrible. I mean, they rated it okay. So I don't think it was a problem with the patient perception of the information. It might have been a problem with the information itself because we don't have a whole lot of guidance. Okay, so what can we say from this trial? So in medical users, medical marijuana cards were associated with developing CUD symptoms, no significant improvement in pain, anxiety, or depression. Cards were associated with improved sleep quality in the short term. Urine assays showed that people can't necessarily trust their product labels, and Ryan Vandry has shown that also by actually analyzing products. I think the results of this trial warrant further investigation of benefits of medical marijuana for insomnia and risk of medical marijuana for CUD. So that's the take home, I think, from this series of publications on this trial. Now for my last 10 minutes, I'm going to completely change gears, and I'm going to talk about driving, because in thinking about risks and benefits of cannabis, this is one that I think people are starting to take more seriously, and that's the risk of driving while high. So in this last few minutes of the talk, I would like to tell you about some studies we're doing using brain imaging to identify THC impairment. Okay, so first of all, what is the true crash risk of cannabis? This is actually a more challenging question than you would think. We know that cannabis in the lab impairs psychomotor skills, divided attention, lane tracking, cognitive function. But when you look at the epi literature, the odds ratios for THC on crashes are relatively small, maybe a 25% increase in crashes. But I would like to argue that we don't have very good epidemiological tracking of this because we don't have a good test for impairment. So if you count everyone with a positive urine screen for cannabis as impaired, your denominator gets really big. Okay, so this is just a map of driving laws. I don't need to get into this, but take it from me, laws are all over the place. People don't know what to do with this. Most states don't even have cannabis-specific drug driving laws because this is just a tough topic. The model for alcohol, like blood alcohol concentration, doesn't work for cannabis because it has unique pharmacokinetics and pharmacodynamics. So THC really peaks in about 10 minutes post-smoking in breath and blood, as you can see here. So THC concentrations go up and then go back down. But when you look at, how the self-reported high window, it can be 2 to 3 hours. So even though your blood can return to baseline, you're still feeling high. And then the problem with a lot of this other testing, like urine or different types of blood tests or even hair, is that you can detect metabolites, not THC itself, but its metabolites for days or weeks or even months. So the window is really hard to boil down in cannabis impairment. And this is a paper showing that people have poor insight to their own impairment. So this is pre-smoking, and this is their drive score. This is worse. So you can see 30 minutes and an hour and 30 minutes, they have a pretty low score, but their perceived impairment goes down. So this is the period where we need to worry about in impaired driving. And to make matters worse, dozens of studies have shown there's no association between THC biomarkers and impairment. This is settled science now. You can't just take a blood level or a breath level and correlate that with impairment. It just doesn't work. Tolerance is huge. This is an issue in alcohol also, but not nearly to the extent that it is in cannabis. So there's a lot of literature out there showing this. Okay, what if we looked at the brain? So when you break a bone, you take an X-ray of the bone. In impairment, the affected organ is the brain. Other methods look at correlates in body fluids, which is a model that we have for drug testing. But what if we just looked at the brain? So this brings me to a technique I've been using called functional near-infrared spectroscopy, or FNIRS, which is a non-invasive technique. This is a little cap on a baby. These are often done in babies, these studies, because they're quiet, they're non-invasive, they're safe, they're portable, they're wireless, they can be used in natural environments. And basically, I'm not going to get into how FNIRS works, but basically, it works on the same principle as fMRI, where neural activation can change your blood flow, which changes the ratio of oxyhemoglobin to deoxyhemoglobin. And the ratio of oxy to deoxyhemoglobin allows for the measurement of brain activity. This is the first breathalyzer. And this is what we have now, something the size of a cell phone. Same thing is happening with FNIRS. So that cap looked really dorky and weird, but I think in a few years, FNIRS is also going to be a small, sleek-looking instrument. Okay, so that brings me to this study. So participants were 169 weekly or more cannabis users. They had 2 visits, 1 week apart, one for THC and one for placebo. We took FNIRS recordings during a working memory task before they got their dose. At 100 minutes after they got their dose, and in this study we used dronabinol, so there was a long period where they were impaired, and 200 minutes after, we had a real life cop, a drug recognition expert, come into the lab and assess them. And then we compared the drug recognition expert to the brain imaging. We used individualized dosing to achieve impairment. And this was really important because we gave people anywhere from 5 to 80 milligrams. With commercialization of cannabis, people have become really tolerant and people weren't getting high. So we ended up increasing the dose and we got IRB approval to give up to 80 milligrams of THC. We dosed them based on self-reported use of cannabis and particularly, like I said, we asked for use patterns, any adverse events they've had, and so forth when we did the dosing. We estimate that about half got impaired. So after we increased the dose, we did much better. Okay, So first, let's talk about these DREs. You're not going to like this. About 20% of people on placebo were judged impaired by the DREs. So the DREs didn't do great. One DRE had 92% accuracy. He was awesome. And one had 50% accuracy. So there was a lot of variation in these folks. So we had to decide if people were impaired. So we had sort of this clinical consensus where blinded RAs would say whether they thought they were impaired or not. And then we looked at their heart rate and their self-reported high. And this is just an example of somebody who did get high with 30 milligrams of THC. Their DEQ, drug effects questionnaire score, went way up, and so did their heart rate. And this is someone on 30 milligrams who just didn't get high at all. And this is their placebo day. So we really were able to distinguish. These are some graphs of the two groups. So this is the group that got impaired. This is their self-reported high. This is the group that we called not impaired. So they got a little impaired, but not drastically. And this was our placebo group. And this is the heart rate. Look at that nice heart rate bump in our impaired people versus our not impaired people. THC doses were not different. So that's important. Oh, I apologize. My phone is ringing. I don't know how to make it stop. Okay, but moving on. So we did an MBAC task, and we wanted to see if groups measured in FNIRS measures. And this was just our cap. We had 5 regions in our cap. Okay, so what we found was there was increased activation in the brain after THC only in people who were impaired. So this is the scan at baseline before the drug in blue, and this is the scan after the drug in red. And this is a significant difference. So remember, both of these groups got THC. Both of these groups got the same dose of THC. But these are the people who didn't act impaired, didn't say they were impaired, and this is the group that did. So what we think is this can be sensitive to actual impairment and not just exposure itself. So the next thing we did was some machine learning on this, because that's sort of the hot thing to do these days. And we wanted to see if we could do individual prediction. Psychiatry has struggled with diagnosing any psychiatric disorder in an individual based on functional brain data, but maybe impairment is such a drastic change in brain function that we could do it. So what we found was we were able to diagnose impairment using FNIRS, that's orange, with about 76% accuracy, which is a little more accurate than the DREs, which is in blue. Our positive predicted value of those who we called impaired, who are actually impaired, was way better than the DREs. We had a 70%, they had a 37%, and our false positive rate was much lower. We had about a 12% false positive rate, and they had about a 35%. So we beat the DRE. We were very happy about that, just looking at the brain. Okay, and for my last slides, I just want to present, this was a working memory task. And so what I've been told is, oh, you know, people aren't going to do a working memory task at the roadside. So we wanted to see if this was possible just using a resting state scan. Can we look at the brain during resting state when they don't have to do a cognitive task? And resting state is a method where you look at patterns of temporally correlated but spatially distant brain activity. And we got basically the same results. So these are the people who never got impaired from THC. This is before they got the drug, and this was after. And these are the people who got impaired from THC. This is resting state activity before the drug, and this is after the drug. And this is the placebo-controlled data. So the other was pre-post data. So people who didn't get impaired on THC and placebo, and people who got impaired on THC and placebo. So you have this dramatic attenuation of resting state connectivity. So what can we say from this study? So prefrontal cortical activation collected with portable FNIRS appears to be a biomarker of THC impairment that could maybe be collected in the field someday. This biomarker is different only in those who are impaired. It's the first biomarker of impairment from THC rather than exposure to THC that I know of. This biomarker can be detected when people are doing a task or when they're at rest. And in future directions, we are going to submit a grant with the MIT Aging Lab to incorporate a driving simulator so we can actually look at driving. That was a whirlwind. Okay, so in my last minute, and then I'm hoping to take questions, I just want to tell you about future directions, 2 studies that are ongoing, which I think are cool. In one study, we're looking at medical cannabis, pain and opioid use in patients on chronic opioids for chronic non-cancer pain. We are randomizing people similarly to what we did in our medical cannabis trial that I presented to a waitlist control or a cannabis condition. And their goal is to taper their opioid dose 10% per month for 6 months. And we want to see if cannabis helps, hurts, or doesn't affect the ability to taper opioid medication. And just a spoiler alert, it's really hard for people to taper opioid medication. So we're not seeing a whole lot of tapering in either group, but we will see. Concerns about pain control, low confidence and ability to reduce their opioids really are persistent. And then this is super cool. So this is a clinical trial where we're looking at cannabidiol or CBD for reduction of brain neuroinflammation in people with chronic pain using PET scans. CBD is implicated in neuroinflammation in some animal studies. So we want to see if we can see a difference in the brain at 4 weeks. And what we're looking for is a reduction in this red, which is a biomarker of inflammation from baseline to post CBD. This is a patient, which I thought this patient looked great. I don't know what the results of the trial will be, but this patient looked great. And we want to see if we can detect an effect. So I am going to stop here and thank all the lovely people who have done the work and have helped me with this research. And again, thanks, Dr. Cooper, for inviting me to give this talk.
That was great, Jodi. Thank you so much. And we do have a lot of questions here. And let's see. I have questions too, but I'll reserve those for later. We have a question from Nico Masali, who asks about during your waitlist study, which pain parameters were you using to monitor pain? And were people also asked about how bothersome or, an affective component of their pain?
Yes. So we used the BPI. And the single item for the daily diary was just pain intensity, but we looked at everything. We looked at pain catastrophizing, we looked at bothersomeness, we looked at everything. I was really hoping to see something, but we didn't see differences in any of the pain measures. And we're using pain bothersomeness in the CBD trial because we think if anything's going to move, it's that. But sadly, we didn't see anything for any of our pain measures.
Did you have any measure of increased function, during the day or anything like that, or quality of life?
Yes, we did. So the one positive finding that we got was perceived stress. So perceived stress levels, we used a perceived stress scale, and that was significantly different between groups. And it was better in the cannabis group than the waitlist group. Other quality of life measures, we had the SF-12, we didn't see anything there. For physical, we saw nothing. For mental, we saw a little improvement, but it wasn't significant, so I didn't talk about it.
All right, thank you. And then Jennifer Pike asked, for that particular study, how did you measure sleep? What scale did you use?
We use the Athens Insomnia Scale. So it's just a basic insomnia scale. I think it's 8 or 10 questions. And then for the daily diary app, we just asked, how is your sleep quality? Because we wanted, since they were doing it every day, we wanted to keep it really focused and quick.
Great. And then Colin Price says, great talk. In real world RCT with MMC, what a cool study, were participants assigned to one group, mood, sleep, or pain for analysis, or could they be in multiple groups based on their symptoms?
That is such a good question. So we asked them, what's the primary reason for wanting to use medical cannabis? So not what's the worst thing. We said, why are you getting this medical cannabis card? I did an analysis where people could be in multiple groups and nothing changed. But you're right, nobody just, especially pain, nobody just has pain. The pain, depression, insomnia are also highly comorbid. But the way that the primary analysis was done was just asking the patient to self-report what they wanted to use the cannabis for.
Great. And then Kathy Kyle asked, can you confirm, and I think you might have talked about this before, can you confirm you needed the medical card even for just CBD? Do you know how many use CBD versus THC for pain or mood disorders?
So it's funny. So this is in the JAMA Network open paper that I published about a year before the main trial. We didn't have a whole lot of CBD users. No, I don't think you need a card. I think you can get CBD at the drugstore now. But there weren't very many people who only used, CBD or CBD oil. Most people actually used high THC products, which was not quite what I was expecting, or 50-50 products.
Yeah. One question I had was looping in the first part of your talk to that part. Were you able to look at differences with respect to people who initiated their cannabis use before 16 versus after 18, and if there were any differences?
Oh, I haven't looked at that, but I should. That's a good idea.
I was thinking also with your Effner study, just
yeah.
And then Connor Murray asked, was past performance in the working memory task more or less accurate than DRE and Effner's in detecting impairment?
Oh, good question. Sorry if something in my eye. I'm not crying. You're not making me cry yet. So the working memory task, the NBAC task, was not a great indicator of impairment. There was a lot of variation, and I think it was too easy. Everybody did very well on it. So I don't think the NBAC task is really designed to be an impairment task. We chose the NBAC task because it elicited prefrontal activation, and it was widely used in the Effner's field. It wasn't really chosen because that would be a good marker of impairment. But I bet it did better than the DREs, because the DREs did really bad.
Within that study, wondering what the public health messaging would be from the results. I mean, the fact that some people were being exposed to 30 milligrams of oral THC and not showing any impairment, no increases in heart rate, minimal intoxication. How do how do you frame those results to the public?
So I had a reviewer who didn't believe it. He said, there's no way you gave people 50 milligrams of THC and they didn't get high. That's what he said. And the only answer I can give is this is commercialization. We have virtually eliminated low THC products in Massachusetts. If you consider low THC under 10% THC and high THC above 10% THC, these products, and these are people who used edibles and waxes and oils, and they've just become so tolerant. And I think that's really why we had to go to our IRB and ask permission to give 80 milligrams of THC. And not everyone got, like you saw that the average dose was 30 milligrams. But with a big standard deviation. Public health messages, we've created really potent products and people are quite tolerant to them.
Great. And then we have one last question from David Cohen, who asks, in the second RCT, looking at effects on pain, insomnia, and mood, did you control for effects of psychiatric medications?
We didn't control for that now.
Also,
we looked at that. It's really hard to look at, from an analytic perspective, because there's so many types of medications and your sample sizes get really tiny. But we tried to see if there was a change in medications, and there was not a change in medications in either group.
Any implications of your concept of impairment on cannabis for the evaluation of subjective effects of psychiatric medications?
Oh, I don't know. So are you saying that for the concept of impairment on cannabis for the evaluation, are you saying can you use Effner's to assess?
So the third question is also if frontocortical activation marks impairment, any implication for activation findings among users of psychiatric medication. So essentially,
we actually looked at that. We actually looked at subgroups of people because we thought, if this is going to have a real world implication, we don't want somebody on Prozac to be called high. And we didn't see any effect on prefrontal cortical activity of anyone on any antidepressants or cold meds or anything like that. Of course, we didn't test every medication, but we had no exclusions in the study. It was not a clean sample. We let everybody in, and then we were able to look at some of that. And I actually have a lot to say about that in another venue. I think being more inclusive is way better in these trials than being super pure, because that's not the real world.
Right. That was great. Thank you so much. I'll speak on behalf of all of us here. Oh, wait, we have one more question. Don Tashkin, what form of cannabis did most of the participants in your study use? Vaping, smoking, edibles? I think
so vaping was number one, followed by edibles, followed by smoking.
Wow.
And a lot of
interesting.
these people didn't want to smoke, and they told me that. They were like, "Ah, I don't want to smoke." So yes, vaping was definitely the most common, probably 2 to 1 to the others.
Wow. That's great. Good. Thank you so much, Dr. Gilman. Maybe one day we can have you over here in person.
That would be so fun. I love California.
Yeah. It's a good place. It's a good place. Great. Well, thank you all so much. And we're looking forward to seeing you at our next grand round and hoping that you can make our journal club later on today. Thank you so much.
Bye, everyone.
Bye.